Alright, so we're going to start up now with our last section on cell division and wrap up by talking about the details of meiosis and also a little bit of what could happen when things don't separate the way they're supposed to during the process of meiosis. So prophase 1 is going to look in a lot of ways, really in most ways, very similar to prophase of mitosis in that we're going to see a cell with the nucleus beginning to degenerate, chromosomes condensing, and a mitotic spindle forming. So really, right off the bat, not a whole lot different from what we saw back in mitosis, but we're also going to see, in this part of meiosis, synapses take place and also crossing over. All right, so remember, again, at this point, we still have pairs of homologous chromosomes, right? So each chromosome is made up of sister chromatids, and they're also going to be paired up. Those pairs of sister chromatids are paired up with their homologs as well during synapses. And during synapses, this is when crossing over can happen. And so in a couple of slides here, maybe on the next slide, we're going to see what exactly that looks like, but just kind of bearing in mind that this can occur with any and all the chromosomes. It doesn't necessarily have to occur. Synapsis always occurs, but crossing over may or may not happen between homologs. It just kind of depends on the luck of the draw. We don't really even fully understand what triggers crossing over or how much of it happens or even whether or not it doesn't occur. It seems to be a fairly random process as far as how much crossing over actually happens during prophase 1. So crossing over, like I said, looks like this. If we imagine maybe this is chromosome, pick a different number this time, chromosome 8. And so here we have dad's chromosome 8 made up of two exact copies of that dad's chromosome 8, sister chromatids, right? We have mom's chromosome 8, also right now made up of two sister chromatids. So we know that this has already gone through DNA replication because now we have two identical copies of all the chromosomes in the genome. So what happens during synapsis is that dad's and mom's chromosome 8s line up together, and potentially at least what can happen is there's going to be a crossing over event that occurs which switches part of mom's chromosome number 8 onto dad's and part of dad's chromosome number 8 onto mom's. Now notice we're just doing it with this one arm of the chromosome, but we could actually do it with these two, or these two, or these two, or any combination, or all of them, or none of them. It's a very, very random process, but in this particular instance, what we get in the end is a chromosome 8 that's entirely dad's. We get a chromosome 8 that's entirely mom's. We get a chromosome 8 that's mostly dad with a bit of mom's, and a chromosome 8 that's mostly mom's with a bit of dad's on it. So if we think about this, for this particular chromosome 8, which eventually each one of these is going to go into one different cell, so 4 separate cells total, every single chromosome 8 that goes into those 4 cells is going to be different from every other chromosome 8 that we see. All right, so metaphase 1. Really not too much different here from metaphase and mitosis, but the one thing that we need to pay attention to here is that in this case, in metaphase 1 of meiosis, the homologs are going to be paired up on the metaphase plate, not the sister chromatids. So, and again, in the next slide here, we're going to see a picture which compares metaphase of mitosis to metaphase 1 of meiosis, and we're going to see, more of a visual representation of what this looks like, but the key thing with metaphase 1 is that the homologs are lining up on the metaphase plate, not the sister chromatids. The sister chromatids are together on the same sides of the metaphase plate. All right, so we'll see, again, like a picture to clarify that here in a second. Metaphase 1, the homologs are lined up side by side on the metaphase plate. And again, this is where that random aspect of the independent assortment takes place, is because the way we line up those chromosomes on the metaphase plate is entirely random. So in this particular instance, we have chromosome 1, mom's on the left, dad's on the right. Chromosome 2, dad's on the left, mom's on the right. But we could do it any other possible way. As long as we have a chromosome 1 on the right and left and a chromosome 2 on the right and left, doesn't really matter what color they are, what parent they came from. As long as we have one of each chromosome on either side of the metaphase plate, then it's a perfectly viable combination. So again, during meiosis 1, this is what it looks like. You can imagine the metaphase plate running right down through the middle of this pair of sister chromatids. Eventually the two red ones are going to go left and the two blue ones are going to go right during anaphase 1, but for right now they're just paired up on the metaphase plate, and during anaphase 1 they're being pulled apart. Whereas in mitosis, when we saw anaphase was involved in separating the sister chromatids. Here, this anaphase 1, is involved in separating the homologs from each other. Now, looking at mitosis, this is what it looks like here. With mitosis, we have, again, this chromosome, pair of sister chromatids lined up on the metaphase plate. So eventually what will happen is this blue one will go left, and this blue one will go right, and that's where they're going to separate during anaphase. But pay attention to the sister chromatids here. Here, the sister chromatids haven't separated. Over here, they have. Over here, the homologs are separating, but over here, the sister chromatids are separating. All right, so again, if we take a look at a simpler kind of hypothetical species that only has maybe 6 chromosomes, so we've got long blue, medium blue, and short blue, and the same thing for red, that's 6 chromosomes total. When we go into meiosis 1, there are 8 possible ways to line up those chromosomes on the metaphase plate. Remember what I told you before? There are 2 to the nth power number of combinations of ways to line up these chromosomes on the metaphase plate. So 2 to the 3rd power then is 8. So we could have all blue ones on the left, all red ones on the right, all red ones on the left, all blue ones on the right, or any other possible combination. As long as we have a chromosome 1, 2, and 3 on the left, and a chromosome 1, 2, and 3 on the right, doesn't matter what color they are, and that's one of the big places where we get some of this genetic diversity during meiosis. All right, so from there we're going to move into anaphase 1. With anaphase 1, we're actually now going to break the chiasmata, which is where the homologs were attached during synapsis and crossing over. They were attached at the chiasmata. They've been maintained all this way up until anaphase 1, and this is the point where the chiasmata is going to break, and the homologs are going to be able to move apart from each other. So in this picture up here, we have the long blue ones moving apart from their long red ones, and the short blue ones moving apart from the short red ones. If we think about how many chromosomes are represented here, there's only 2. Look at how many chromosomes are represented here. Also, there's only 2. So again, the colors don't really matter for all intents and purposes. As long as we have a long one and a short one on each side, then that works. All right, so we're moving the homologs apart from each other toward opposite ends of the cell. By the time this is done, each end of the cell is going to have a haploid set of chromosomes. So remember, this cell started off with 4 chromosomes: long blue, long red, short blue, short red. At the end of this, up here at the top of this cell, there's going to be long blue and short red. That's it. There's only 2. Down here, there's going to be long red and short blue. And again, that's it. Doesn't really matter that we still have chromatids attached here. That doesn't count. We're just looking at numbers of homologs at each part of the end of the cell. So even though, yes, we still have chromatids attached down here, this still is only representing 2 chromosomes, as is the top of the cell only representing 2 chromosomes. And that, like I said, is that independent assortment that we talked about with that random lining up of chromosomes on the metaphase plate. All right, telophase 1. Not really a whole lot to say here, different from mitosis. We're going to see the nuclear envelope come back. We're going to see the breakdown of the mitotic spindle. That's also going to happen here as well. We're not necessarily going to see decondensation of chromosomes because in a lot of cases, at least for males, as soon as these cells are done going through meiosis 1, they're going to jump right into meiosis 2 and keep going. Females, different story. Female gametogenesis is a very different kind of a thing, but in males, these 2 cells that are produced are automatically going to go immediately right into the second round of meiosis. So the chromosome decondensation thing doesn't necessarily happen like it does with mitosis, but the other 2 parts do take place. So we're going to have cytokinesis. These 2 cells are split apart from each other, and then eventually meiosis 2 can happen. And again, this meiosis 2 interval here, this is a very, very different thing for males versus females. Like I said before, males jump immediately into meiosis 2, but for females, meiosis 2 only occurs after fertilization. All right, so in females, like literally the number of cells in your body that have ever gone through meiosis 2 during your lifetime will equal the number of conceptions you've had, because that is the only time when meiosis 2 occurs is after a fertilization event or after conception. And so there's a lot more detail we could go into that if we were maybe in a different class. In Principles 1, we don't have quite that much time to go into it because it's kind of a deep topic. But again, if there's questions about this, please make sure you ask me either during WebEx or through email. We can talk about that more as far as what exactly happens to the cells that are produced in meiosis for females, when it takes place, how it takes place, that kind of thing. But it's a very, very different thing for males versus females for this whole meiosis 2 interval thing. Now, meiosis 2, I told you back in the last lecture, is really just the same thing as mitosis. So if we kind of break this down, I'm going to read through it really fast here. But prophase 2, what happens? The nucleus goes away and a mitotic spindle forms. Again, we didn't really decondense the chromosomes, so there's not really a need to recondense them here. But that's basically the same thing as mitosis taking place. Metaphase 2, sister chromatids line up on the metaphase plate, just like mitosis. Anaphase 2, sister chromatids separate and move apart from each other, just like anaphase and mitosis. Telophase 2, in cytokinesis, we end up having 4 cells, each with its own set of chromosomes, each with only a single chromatid, just like it occurs at the end of mitosis, although in mitosis we only make 2 cells, but here we have 2 cells making 2 cells each, so 4 cells total at the end of meiosis 2. All right, so meiosis 2, I go through it really fast because if you know mitosis, you really kind of sort of know meiosis 2 by default. Meiosis 1 is the part that's really kind of quite a bit different from either mitosis or meiosis 1. So the end result of meiosis is a grand total of 4 cells, each of them being haploid, each of them only having a single set of chromosomes, versus mitosis where we create 2 cells that have the exact same number of chromosomes as the parent cells had. So there's a big difference there. Now in males, each of these 4 cells can become a sperm cell. In females, only one of those cells could even potentially become an egg cell, and that again only happens if there's fertilization. The other 3 cells that are resulting from meiosis become something else, which is actually not used in any way for reproduction. They actually are just broken down by the body. And again, that's one of those topics where we could go into a bit more detail if you want, but for the purposes of this class, we don't really have the time or scope to go into it with this course. But again, if you have questions about it, I'm more than happy to answer those as thoroughly as you'd like me to. In animals, these are the cells that become the gametes, so either sperm cells or egg cells. If we're dealing with plants or fungi, some of the protists, the cells that come out of meiosis 2 then go through several rounds of mitosis, and what that does is to help them amplify or multiply the number of gametes they produce. So if you think about plants that start to produce pollen, for example, pollen is created by the hundreds of trillions of granules in plants. They go through a few rounds of meiosis, but then most of the gamete production for them is just recopying those same haploid cells over and over and over again to give them a greater number of gametes they can use to actually hopefully successfully do fertilization. So it's a way, mitosis is a way for them to kind of like ramp up the numbers they're going to produce before they start releasing those into the environment. All right, so what about some errors that take place in meiosis? The term non-disjunction refers to a situation where something doesn't separate the way it's supposed to. If something is disjunct, that means it's separated, it's dispersed, it's not really connected. If non-disjunction occurs, that just means it's not coming apart. All right, so during meiosis, if something doesn't come apart the way it's supposed to, we say that a non-disjunction event has occurred. All right, so this can occur either during meiosis 1 or meiosis 2, but when it occurs, it results in the production of what are called aneuploid gametes. So aneuploid gametes are those that have, in humans, maybe you've got 22 chromosomes or 24 chromosomes. That would be an aneuploid gamete. Or 21 or 25 chromosomes would be aneuploid. It's not that you have a whole extra set. There's a term for that as well that we'll talk about here in a few chapters, but just having a whole set plus or minus 1 or 2 chromosomes is an aneuploid gamete. And so the most common outcome of fertilization when aneuploid gametes are involved is that the zygote that's produced will die almost immediately, because having extra chromosomes or missing chromosomes is not a way to develop successfully. You gotta really have just the right number of chromosomes to be successful as an offspring. So typically when this occurs in humans, and we all make aneuploid gametes throughout our lifetime, the likelihood of any of us producing an aneuploid gamete that would actually be involved in fertilization is very, very low. But if it does happen that an aneuploid gamete is involved in fertilization, typically the pregnancy is over before it even begins. So person doesn't even realize that anything happened. There's no, outward sign of pregnancy. There's no development whatsoever. The zygote forms and either dies as a zygote or maybe dies after a couple of rounds of cell division, and nobody even knows that anything happened because it's over so quickly. Right? Having the extra chromosome or chromosomes or missing chromosomes is usually pretty catastrophic for animals that have that particular situation. The exceptions are situations like Down syndrome or trisomy 21, or situations where we have extra sex chromosomes. We'll talk about more in a couple of chapters. There's a couple of other times when you can have an extra chromosome, but they're really, really pretty uncommon for anybody to survive having too many chromosomes or not enough chromosomes. So the last little bit here that I want to talk about, and we will talk about non-disjunction more in the next chapter, at the end of the genetics chapter. The last little bit of this chapter that I want to focus on is just kind of comparing mitosis to meiosis. All right? So things that are unique about meiosis are as follows. Crossing over and synapses. That is definitely not something that ever occurs in mitosis. Crossing over and synapses are absolutely unique to the meiotic process. And so the big benefit of that, again, is that it creates genetic diversity among the offspring, in addition to all the other sources of diversity that we'll talk about here and kind of summarize in a second. We also have the fact that sister chromatids remain together throughout the entire process of meiosis 1. In mitosis, the sister chromatids are separated as part of the goal of mitosis. Same thing with meiosis 2. Sister chromatids are separated. But for meiosis 1, the sister chromatids remain together throughout the entire process of the first round of meiotic division. This is kind of related to that number 2 point, but the kinetochores, which again are the places where the mitotic spindle will attach to a chromosome, they are going to move toward the same ends of the cell for the sister chromatids during meiosis 1, which just means the sister chromatids move together throughout meiosis 1. That's also kind of unique to meiosis. And then finally, we don't have any DNA replication between meiosis 1 and meiosis 2, which is unique. Because again, if you had that, it would actually completely defeat the purpose of meiosis by having this extra round of replication. Remember, the ultimate goal of meiosis is to make cells that have half the number of chromosomes. If we redoubled the chromosomes in between meiosis 1 and meiosis 2, that would take us right back to where we began, thereby completely defeating the purpose of meiosis. All right, so just a quick overview here of mitosis. And something else that I always like to put in a pitch for, and when I teach this course in person, I always do this. Try to draw this out. All right, get some crayons, get some colored pencils, draw some homologous chromosomes, draw some sister chromatids, and draw each of the steps of mitosis and meiosis both. Don't try to do it for humans. Don't try to do 46 chromosomes, but try doing maybe 4 or 6 chromosomes total and work through the different steps. Maybe even write down the steps that occur under prophase, metaphase, anaphase, telophase as you're drawing through this. And it's a really, really good way to help you remember exactly what's going on during each step of the way. Meiosis, a little bit more complicated, but it is basically the same thing happening twice. And so again, a good way to work through this is to practice drawing it out on a piece of paper and kind of keeping up with the chromosomes and what's happening where, what's produced when, and that will help you kind of learn this process as well. All right, so just a couple more points of comparison here between mitosis and meiosis. The other thing I want to summarize really quickly that I don't have on a slide is where genetic diversity comes from through sexual reproduction. All right, so sexual reproduction generates diversity basically in 4 ways. Number 1, you've always got mutation. That's always a way to get diversity. Now, it's not a very common way, but it is a way to sometimes get some diversity in the offspring. Number 2, with sexual reproduction, you have genetically unrelated parents producing offspring, which is again another way to create some diversity among the offspring. Number 3, we've got crossing over. And number 4, we've got the independent assortment thing where the way the chromosomes line up on the metaphase plate is an entirely random process. So those 4 features together are how we get diversity out of sexual reproduction. Now the last little bit that I have here, and this is something which I've added relatively recently in Teaching Principles 1, is a really, really quick overview of spermatogenesis and oogenesis. So in other words, male gamete production versus female gamete production. The way this starts off for males, simpler process here by far. Spermatogonia are the starting cells. These are the ones that we called germline cells back at the previous lecture. They go through mitosis throughout the life of a male. This starts at puberty and continues until a male dies, albeit maybe at a lower level, not quite as quickly or as efficiently, but it does occur throughout the lifetime of males. So spermatogonia are always replacing themselves. Some of those spermatogonia, the ones that don't continue the pattern of doing mitosis, they're the ones that are going to go into meiosis. So some spermatogonia stay spermatogonia and keep dividing through mitosis. Other spermatogonia differentiate themselves into primary spermatocytes, which then go through meiosis 1. Now these cells are still diploid. In humans, they still have 46 chromosomes, but they go through meiosis 1 and they come out having 23 chromosomes. They come out being haploid. So the secondary spermatocytes are the result of meiosis 1. They're haploid. And they're the cells that go through meiosis 2 to produce what are called spermatids. Now, spermatids are, genetically speaking, perfectly viable as sperm cells, but in terms of, logistical terms, they're not quite there yet. Spermatids have to go through a differentiation process to become sperm cells, which are then are able to go through and fertilize the female gamete. So it's going to look something like this. Spermatogonia, some of them go through mitosis continuously to make more spermatogonia. Others are going to become primary spermatocytes, which then go through meiosis 1 to become haploid cells. So we started off with 4, now we have 2. These 2 secondary spermatocytes now go through meiosis 2 to become spermatids, which now have one chromatid for each chromosome, and they are again haploid, so these only have two chromosomes because the parent cell had four. Each one of these four cells can then differentiate into a functional sperm cell. Now, for females, even without getting into all the details of timing and hormonal control and those kinds of things, which I guess we didn't do for males either, but for females, it's a more complicated process. It starts off similar, but the timing is very different. All right, so this early process actually happens well before a female is ever born. For those of you female students in the class, you did this before you were born by many, many months. All right, this is a really, really early part of your biology where your oogonia initially go through mitosis for a while, and again, some of the oogonia become primary oocytes. They're still diploid. They're the ones that go through meiosis 1. And at that point, we have what are called secondary oocytes. So at the end of meiosis 1, there is a single secondary oocyte produced and a single polar body produced. Now, the secondary oocyte and the polar body are both haploid because they both went through meiosis 1, but the polar body is the part that's actually broken down or degenerates relatively early on. The polar body is essentially just like a garbage can for chromosomes that aren't used for anything. So this first polar body, eventually what it does is actually goes through a second round of meiosis, and these two polar bodies then degenerate. The secondary oocyte is then put on sort of like a holding pattern, suspended animation kind of a thing. This secondary oocyte goes into dormancy, more or less, and it stays there. And it stays there until a female achieves puberty, which at a minimum is somewhere between about 11 to 14 years for a human female. Those secondary oocytes have been sitting since before you were born, all the way up through the beginning of puberty, at which point the secondary oocytes begin to be ovulated at a rate of around once per month. All right, so those secondary oocytes are what actually occur or are released during ovulation, which can then be fertilized by a sperm cell if there are sperm cells pregnant or if sperm cells present. Then pregnancy happens. So secondary oocyte, if it's fertilized, and only if it's fertilized, the secondary oocyte will then go through a second round of meiosis to create an egg, and also a third polar body, which then also breaks down like the first and second polar bodies did. So again, extra chromosomes left over, they didn't make it to the egg cell, they go into a polar body, this thing breaks down. The egg cell, and by the way, the egg is really only ever produced after fertilization. We typically say that an egg is ovulated once a month. It's not technically speaking an egg, it's actually a secondary oocyte, but egg is a lot easier to say, so most people just go with that. But the egg actually officially forms after fertilization and after meiosis 2 takes place. There's an egg very, very briefly before it becomes a zygote. So that's female gametogenesis. I'm not going to ask you a whole lot about that for this class, but we can certainly talk about details more if you want to. If you have questions about anything, that's perfectly fine as well. Feel free to ask. But for now, we're going to wrap up our chapter on cell division, and we're moving next into our chapter on genetics.